Hey guys, it's Mrs. Olenichek, and today we're going to talk about proteins. So proteins are one of the more structurally and functionally diverse groups. So they have lots and lots of different functions, and they're involved in pretty much everything. Some examples of functions are enzymes. They can be structural. They can be important carriers, so moving things around your body, like hemoglobin carries oxygen and carbon dioxide, or they could be transport proteins, so they allow for materials to move in or out of the cell. They're important for communication. They can be the signal itself, or they could be the protein that receives the signal. They play roles in defense, movement, even storage. So when we look at their structure, the monomer, so proteins are polymers. They're made up of long chains of amino acids. And those amino acids get put together to form this polypeptide chain. There's 20 amino acids, so 20 ingredients that are found in proteins. So the monomer is the amino acid. The polymer is a polypeptide. So that's basically a long chain of amino acids. Proteins can actually be made up of multiple polypeptide chains, so some of them are just one polypeptide that gets folded in on itself, but there's others that actually need to have multiple polypeptide chains come together to make them function. And here you can see some examples of different proteins, and they're all kind of folded in on themselves. So here you have some growth hormones, here you have hemoglobin, we'll see this guy a bunch today, and this has 4 different polypeptide chains coming together, and here's RuBisCO. So if we look at the amino acids themselves, they're made up of a central carbon, an amino group, a carboxyl group, and an R group. So amino acid, so you've got the amino, and then the carboxyl, COOH, which is the acid part, and then you get the R group. And the R group is really what gives it its properties. It's going to be variable, so if there's 20 amino acids, there's 20 R groups. And so each amino acid has a different R group, and that R group is what's going to give it its kind of function, how it acts. And so you can have amino acids that are nonpolar. So if you look at these guys, they've got kind of nonpolar side chains, and so they're going to be nonpolar and hydrophobic. Now, don't worry about memorizing all of these, just understand that there's going to be different R groups with different properties, and that's going to influence the structure. So you should understand that they're going to be nonpolar or hydrophobic because of these R groups, because these are nonpolar. That makes the amino acid itself nonpolar. Here you also have your polar. You can have some that form acids in solution, some that form bases, and then you have these guys. These are really important, the ones that form disulfide bridges. And these are going to be really key in terms of stabilizing the 3D structure. And if you're interested, you can look up what that has to do with perms. So all of those different amino acids, so 20 amino acids, actually results in tons and tons of possibilities for protein structure. Because if you think about it, proteins can be any length and any combination of those amino acids. They can come in any order or have any combination of that order. And so you really have kind of this infinite diversity of proteins, which is why they have so many different functions. So if you look at this, we can vary the length. If we just focus on polypeptides chains that are just 7 amino acids long, that would be 20 to the 7th power combinations. And so if you think that some proteins can be up to 30,000 amino acids long, then you can see why there's so much diversity in structure. All right, so those 20 amino acids come together via peptide bonds. So essentially it's a covalent bond between the carbon on this amino acid and the nitrogen on this amino acid. And in order to get those two to come together, you need to pull off a OH or a hydroxide over here and a hydrogen here, and what you end up with is a water molecule coming out and these two forming that covalent bond between them. We call that dehydration synthesis. Sometimes you'll see it called a condensation reaction because you're pulling a water molecule out and you're forming that peptide bond. So the big idea here when it comes to proteins and their structures is it's the order of the nitrogen bases in DNA that codes for the order of amino acids in a polypeptide. So DNA's job is to hold on to instructions on how to build proteins. And so the order of the bases in DNA, those A's, T's, G's, and C's, determines the order of amino acids in that polypeptide, which determines the structure of the protein, which is really what determines its function. So it's kind of the progression. And if you can remember that, you're in really good shape, especially when we start looking into genetics and how changes in DNA can affect protein function, which is really the traits that you have. Those proteins have a huge influence on the traits you see in an organism. So kind of big picture is your DNA codes for messenger RNA. Remember, that happens in the nucleus. We call it transcription. And then the messenger RNA carries that information out and helps to build the protein. It does that through the process of translation with the help of a ribosome and some transfer RNA. And then that protein is what gives you your traits. So again, big idea, the function is dependent on the structure. So all of these different proteins have very different structures and very different functions. So let's look at how that structure forms, or kind of the layers of that structure. The structures, we kind of divide them into two big categories. You have globular proteins, which are kind of globs, right? That's why they're called globular, or at least that's how I remember it. And they tend to be round-ish and serve more functional roles. And then you have fibrous proteins that tend to be long and narrow, and they tend to serve more structural roles like collagen. All right. Excuse me. So if you go through those structures, and if we look at like how that structure comes to be, how do you become fibrous versus globular? First we look at the primary structure, and that's all about the order of amino acids, right? So that's determined by your DNA, and that's really super important in terms of figuring out the final structure. So that really is what determines everything else after it. And a slight change could actually have a huge influence on how that protein functions. So for example, in sickle cell anemia, hemoglobin, you change one amino acid from a hydrophilic amino acid to a hydrophobic amino acid, and you have a huge influence on the overall structure. So that's primary. Secondary structure has to do with hydrogen bonds that are going to form between adjacent amino acids. So they're right next to each other. They tend to form these things called alpha helix or beta pleated sheet structures. Then you get tertiary structure. This is kind of the big folding of the polypeptide, and this has to do with amino acids that aren't next to each other. You have hydrophobic interaction, so your cytoplasm is going to be water-based, right? You are a big watery environment. And nonpolar amino acids are going to want to get away from that watery environment, and so they tend to cluster towards the internal portion of the protein. Then you get hydrogen bonds and ionic bonds between different R groups in the amino acids, as well as those disulfide bridges, and those are going to be really good for stabilizing the three-dimensional shape. So now you've folded up your polypeptide, and you get to quaternary structure, and this is only in certain proteins. So some proteins need to have multiple polypeptide chains come together, and that's where you get that last layer, that quaternary structure. And that's all about hydrophobic interactions as well. So here, just a quick review, you've got primary structure, which is all about amino acid sequence and those peptide bonds, and that's determined by DNA. Secondary is about the R groups and the hydrogen bonds they form between those adjacent amino acids. Your tertiary level is going to be R groups as well, but it's going to be hydrophobic interactions, disulfide bridges, and you get your hydrogen and ionic bonds between amino acids that are further away from each other. And then last, you get multiple polypeptides coming together, and that has a lot to do with hydrophobic interactions too. So it's kind of a review of the overall way they fold up on top of each other. Now, remember that structure determines function. So if we start messing with the structure, so anything that's going to change the way that that protein folds is going to affect its ability to function properly. And so we say when a protein unfolds, it denatures. If it changes its shape, it's going to denature and it won't function as well. And so any conditions that are going to disrupt that secondary and tertiary structure, those bonds that form, the hydrogen bonds, ionic bonds, or those disulfide bridges, are going to cause this denaturation. So things like temperature is going to disrupt the bonds, pH, you're changing that external environment, and by doing so, you're going to change the way that those molecules or those R groups interact with each other. Same with salinity. You're adding dissolved ions with the salt, right? And so all of those are going to have an influence on whether or not those bonds form and where they form. And so they're going to disrupt that secondary and tertiary structure and alter the shape, which kind of destroys the functionality. Now, some proteins can return to their shape after denaturation, but many can't. And so understanding the conditions that are going to cause that denaturation is going to be really important in terms of understanding how they function and how to adjust function in proteins. And we'll talk more about this when we look at enzymes, because really anything that affects the enzyme structure is going to affect its ability to function. All right, here's some of the proteins that you need to know for IB. It's always good to have a few of these kind of in your pocket to make sure that you can describe them. We've got RuBisCO. This one's an enzyme. It's super important for photosynthesis. Its real name is ribulose bisphosphate carboxylase, or RuBisCO. Insulin, a hormone. It signals cells to take up glucose, which helps lower blood glucose, and it's secreted by the beta cells in the pancreas. Immunoglobulins, these are also known as antibodies. They're really important for your immune function. They're going to bind to pathogens and get them to stick together. They also act as a marker for phagocytes, telling them to come over and eat the pathogens. Rhodopsin is going to be a protein that's important for vision. It's found in rod cells, and it's important for light sensitivity. Collagen. Collagen has a ton of different forms, but they're all rope-like proteins, and they actually make up about 1/4 of all the proteins in the human body. They're important for skin and elasticity. They give strength to tendons and ligaments. They form part of teeth and bones, and so they're going to be really important for all of those structural roles. And then last but not least, we get our spider silk, the spiders all throughout the presentation. Spider silk is really interesting. It has there's different types with different functions, but some of them can be stronger than steel and tougher than Kevlar. If you look at their structure, they have these really interesting different structures. They have different regions with different properties that allow them to be really strong, but also resistant to breaking and flexible. So there's a lot of research done into that structure. And that's it for today. So if you have any questions, make sure you ask them when you get to class. We're going to be looking at structure a little bit more closely once you get there. All right, bye.